All right, good afternoon, 7th hour. As you can tell, I'm away at a track meet. All right, so our objectives, once again, are to summarize the role of the enzymes involved in the replication of DNA. So enzymes, once again, remember, are proteins, which are one of the four biomolecules. We have proteins, we have carbohydrates, nucleic acids, and lipids. Then we're going to explain how the leading and lagging strands are synthesized differently. And remember, synthesize means to make. The main idea, DNA replicates by making a strand that is complementary to the original strand. So semi-conservative. So once again, we discuss semi as half and conservative as meaning saving things back for later use. So essentially what we're going to do is take half the DNA strand, so our double helix, we're going to take the left half, for example, and we're going to synthesize a new right half from that. So half of it will be old strand, half of it will be new synthesized strand. And then we'll do the same thing to the right half, and therefore we will have two identical copies of DNA at the end. Remember, the synthesis is done in the interphase, the subphase of S, so the synthesis stage. That's where we will replicate the DNA. You can see from the photo here, once again, using that template strand of DNA, breaking it apart, and adding the nucleotides, and you can tell that we have half old and half new DNA in the end that are identical to each other. Now, how do we do this overall process of replication? Well, as we discussed last time, we have a couple enzymes, and these are the first two that we got to. Once again, we got to unzip the DNA, or basically take it apart, and we can do that with DNA helicase, which is like Chuck Norris at the butt kicking contest. It goes through and breaks those hydrogen bonds between the A's, T's, G's, and C's. Once we have it separated, we got to do something with it. Otherwise, it's going to stick right back together. So another enzyme, RNA primase, will come by and add short segments of RNA to the nucleotide strands. So this will start laying down a few nucleotides so that polymerase can come in next and start adding nucleotides. Alright, so once again we discussed this picture. You can see your alpha helix or double helix. Helicase is going through there, unzipping it, and then the primase would come in right behind it, start putting down some primers so we can get this whole process of replication started. This is another in-depth photo as we discussed yesterday as well. You can see helicase right about here. So you got helicase right here, and it's going to go and travel in this direction, unzipping the DNA, breaking those hydrogen bonds. At the same time, we have primase right here coming in, laying down short little segments of primer so we can start to synthesize the new strands as you see here and also here. Okay, so we will be talking about DNA polymerase next, so hold on. Okay, got it erased. Now we're going to move on. Or backwards. I'm not really sure where we're going now. Yeah, lovely. There we go. Technology, my best friend, said no one. All right, so stage 2, base pairing. All right, so fixing the DNA strands, okay? So DNA polymerase, once again, is an enzyme, but its job is to go out and find the correct nucleotide and piece it together on that DNA template strand. So, for example, if it said like an A or a T, or an A, it would go and find a T to match up with it. If it said C, it would find a G, and it just goes out and finds the right one so we can start to synthesize that new strand. And it keeps in mind the base pairing rules. That adenine always matches with thymine, and thymine and adenine, and cytosine and guanine and cytosine. All right. Now, once again, the nucleotides are attached to the 3' end of the new strand of DNA. So when we look at it, once again, that first original picture, this is a little more in depth. You have the original parent strand, we separate it, and we're going to add the new nucleotides in it. So you can tell that each base pairing rule is abided by so that we get the right sequence and get an exact copy of the DNA. Now, well, as we go along here, the base pairing, we know that DNA is anti-parallel, which means it runs in opposite directions, kind of like cars on a highway. Some are going south, some are going north at the same time. So if you look at the DNA, we once again have our 3' and 5' end. On the opposite, we have the 5' and the 3' down here. So we know they're anti-parallel. The only problem is DNA can only be read from the 3' end to the 5', which means we can go in this direction and in this direction. Now, you're thinking, okay, whoop-de-doo, what's the big problem? It becomes a big problem when helicase can only travel one direction, this way. So the DNA has what is called the leading strand and the lagging strand. So the leading strand just follows helicase right down the line, no problem. But the lagging strand has to always play this catch-up game that never ends. So as the helicase goes, it's plowing the road, essentially. Then the polymerase has to stop, jump back as close as it can to the helicase, and start again, which create these small little fragments right here. And they're about 100 to 200 nucleotides long. All right? And they're called Okazaki fragments. Now, I know, Terrell, you're thinking Chinese. No, this guy was actually Japanese. Okay? So he was the first to discover it, and therefore they named him after him, of his great accomplishments. So these Okazaki fragments do exist on the lagging strand. We are going to talk about how this becomes problematic because in the DNA, as we look, we have these little short gaps right about here. So you do not want to leave gaps in your DNA because it could break off and fade away and then be lost to the pages of history. But we don't want to have missing pieces of our DNA. So what we got to do is figure out how to fix that. So the way we fix this is stage 3. Stage 3 is the joining of the backbone, essentially. So all those little fragments, the Okazaki fragments, they're pieces that need to be repaired before we can actually be, or our DNA can be fully functional. So we have another enzyme, yet again. This one is called DNA ligase, and is an enzyme that will join the disconnected sugar and phosphate backbones together. Now, the way I remember this one is ligase fixes the backbone of DNA. I remember it as at night, you lie down on your back. So ligase fixes the backbone. At night, you lie down on your back. So ligase, back. Okay? In the lagging strand, it is used to connect the Okazaki fragments and any other breaks within the DNA, so the sugar phosphate backbone. Now you might be thinking, there's other breaks in the DNA? Well, keep in mind, there's not just one DNA polymerase in your entire cell. There are several of them. And your DNA is about, if we stretched it out, all your chromosomes from end to end, and that DNA unraveled would be about 6.5 feet long. Okay? So that would take some time to replicate all that DNA. So you have several polymerases actually doing this process of replication in several different areas along the DNA. So even though it's a leading strand, you may actually have to repair the backbone of leading strand to leading strand because two polymerases were working in two different areas and they finally caught up to one another. Okay? So the Okazaki fragments were repaired, no harm done there with DNA ligase. So this is just a recap, once again, a nice little picture that sums it all up. And if you look, we've got our helicase right here, and what it's doing is it's going to break those hydrogen bonds. We've got our DNA polymerase coming in, okay, adding new nucleotides. But the question is, how did it get started? We had an RNA primer, okay? And that RNA primer will lay down that little starting strip, and then it'll get that polymerase going, and it can add more nucleotides as it goes. All right. Now we also have the leading strand, as it says down here, which will follow helicase right down the line. But then we also have the lagging strand that has to play forever catch up, and therefore we have these short little segments here, here, and here. All right. And finally, last but not least, DNA ligase will come in and fix those backbones for us. And that's ultimately what we need, is just two synthesized strands that are identical and fully intact. All right. Now, let me pause it here so I can get rid of the lines. Okay, so that pretty much concludes our DNA replication process. But there are some key differences between replication in you as a eukaryotic organism, so you, plants, fungi, protista, which are all eukaryotic if you recall, and prokaryotic individuals, or like bacteria and archaea, if you remember. So in eukaryotic DNA, your replication, you have basically long linear chromosomes. Your DNA is an alpha helix, starts at point A and ends at point B, essentially. The replication occurs in several places along the length of the chromosome. So once again, you have more than one polymerase. It will start in several different locations. So if this was, say, your DNA strand, we will start polymerase here and say here, and this one will run this direction, and this one will run this direction. And therefore you can have another discontinuous strand right here, even though it's a leading strand. Okay, so that's how your DNA replication goes. Now I can already see some hands going up and think, well, how's this any different than maybe prokaryotic replication? Yep, all right, well, we're getting there. So prokaryotic DNA replication, like a bacteria, their DNA is not linear like ours. It's actually circular, like this. All right, so replication starts in one place and goes in each direction until the replication is complete. So it would start in this direction and also in this direction. So when it's done, it will actually make what looks to be like two little rings stuck together like this, and then they'll just pop off, and one will go this direction and one will go this direction, and then eventually the cell will divide and make two new cells. So that's how bacteria do it. So a little different than ours because their DNA is connected end to end and ours is not. All right. So differences between eukaryotic replication and prokaryotic replication of the DNA. All right. So here's the little video I was telling you about. I'm not going to play it here. You guys can get your Chromebooks out and check it out there. It talks about everything we've talked about in section 1 and section 2. It is a little slow, but if you need to see it in motion, sometimes that helps. Just a nice animated picture that shows how the leading and lagging strand work. That's something you really want to look at. That seems to be something usually a lot of people struggle with, visualizing that. So give it a look at your own time and convenience, but check it out. And prokaryotic DNA model using the following pieces. So look at the straws, you'll see them, they're different colors. The cytosine is C, which is blue straws, thymine is green, adenine is orange, and guanine is yellow. Okay, the phosphates are the white straws, and then the hydrogen bonds are the little white rods in between them. You'll see them. Okay, deoxyribose sugars are the black pentagons with the 3 prongs. And what you are to do is to build this particular model here. So you'll have to go through and code for each one of these. So you have to figure out, and what I would do is I'd write the colors. So if it's A, adenine, you're going to need the orange one up here. So you might just write the colors along the top so you can build your entire strands. And it should look just like this when you're done, okay, according to the color chart at the top. All right. So here are the procedures on what you need to do. You can bypass number 6. You don't have to do that. But when you're done, dismantle it and put it back where you found them. Okay? Now, at the end, you do have a few things to go over. Diagramming the DNA replication. Name 2 differences between eukaryotic and prokaryotic replication, which should be pretty easy. We just went over that. And then do a little matching and then show me that you know how to replicate this DNA down here. So basically matching up the correct nucleotides with the sequence. Okay? Hopefully this works for you. Good luck, and just listen to your sub and whatever they have to tell you. So we'll see you tomorrow, guys. Bye.